All right, well, thank you everybody for joining us here at the NYHIA. My name is Aidan Woishnis. I'm a board member of the HIA. Today we're celebrating Hemp History Week, and we're very lucky to be joined by Larry Smart, a professor of horticulture, plant breeding, and genetics at Cornell University. That's quite a portfolio you have of knowledge base there. We look forward to really hearing all the research that you have to present. I know you've had a lot of experience in the hemp space and a lot of different topics in the hemp space, and half an hour I'm sure will be enough to get a taste of it, but not enough to get the full depth. But anyway, Larry's a leader in the hemp space, and for the next half hour he's going to give a presentation, and then afterwards we're going to have about half an hour for Q&A. So please all queue up your questions, whether you're calling in or whether you're here on Zoom. You can ask through chat. We'll get into that. So Larry, you want to take it away? You can share your screen and we'll get going.
Okay, great. Thanks very much, Aidan, for this opportunity. I'm probably going to go a little bit longer than 30 minutes,
Okay.
but feel free to cut me off if I go too long. But yeah, let's get started. I'm going to focus on a presentation on some of our CBD trials over the last 2 years. Okay, is that looking okay? Give me a thumbs up.
It looks good.
Okay. Yeah, and if there are any technical problems as we go through the presentation, I apologize in advance if my Wi-Fi cuts out, but it's been pretty stable recently. So I want to start by acknowledging and thanking the work of all the folks in my lab, graduate students George Stack and Jacob Toth, and my postdoc Craig Carlson, and technicians Rebecca Wilk and Deanna Gentner. I'm going to show you a little bit of data on powdery mildew that was collected by my wife, Chris Smart, who's a professor in plant pathology, and her PhD student, Ali Kalla. All of our Ithaca trials are managed by Professor Don Vyans and Julie and Jamie and his group. And all of our cannabinoid analysis was done by Joss Rose and his postdoc, Glenn Philippe. So we're going to focus in on CBD production, which occurs in these specialized cells on the female flower parts called trichomes, and specifically in the little bulbous tip of these trichomes. That's where the genes are expressed and the enzymes to carry out that biochemistry to produce CBD and THC. Those are primarily only in the female flowers. So we've done a lot in the hemp world to produce fields with only female plants. I'm not going to talk about that right now, but it's a pretty interesting question why these trichomes and these chemicals are produced on the female flowers in the first place. So let's get into the biochemistry a little bit, and then we'll talk about the genetics behind this biochemistry. So here's a pretty nice diagram of the biochemical pathway for the production of THC, cannabidiol or CBD, and cannabichromene or CBC. All three of those compounds are made from a common precursor, cannabigerolic acid or CBGA, and cannabigerolic acid can be converted into CBG as well. So you see that the first step in the conversion of CBGA is carried out by 3 enzymes: THCA synthase, which produces THCA. THCA is converted through heat to the active delta-9 THC. CBDA synthase, which again likewise produces the acidic form, which is converted to CBD through heat, or CBCA synthase. And we know the least about the CBCA synthase compared to the other 2. And again, CBGA can convert through heat to CBG. As we've been doing, so let me just say that we analyzed these compounds at Cornell, again in Joss Rose's lab in plant biology by HPLC. So most of the data I'm going to be showing you is coming from shoot tip sampling from sort of the optimal part of those female flowers. We dry that tissue at room temperature hopefully not letting it get too hot, mill it in a Ninja food mill, and then extract it with a very small volume of ethanol, and then run it through the liquid chromatography device. And we get peaks like this, where we see the abundance of CBDA and CBD and delta-9 THC and THCA, for example. So those are the predominant peaks out of all of the cannabinoids that we analyze here. We use, just a note, we use standards that we can purchase from a company to know exactly what the concentration is for a peak of this height. We establish a standard curve from known concentrations of these compounds. It is very challenging to establish a standard curve that includes the proper ranges of concentrations for all these different cannabinoids in a single run. And what we found is that as we get to higher and higher levels of CBD in our samples, we tend to be above the highest level of our standard curve, and we lose some accuracy in that. And that will ultimately have some impact in variation in those ratios that you see of total potential CBD to total potential THC. So as we have been sequencing more and more cannabis genomes, we're getting a lot more genomic data now. We are learning more and more about the genes for CBDA synthase and THCA synthase. And as you can see in this cartoon, what we've learned is that those genes sit right next to one another on the chromosome. They are, as we say in genetic terms, they are very tightly linked, and they behave as if they were a single gene, what we call the B locus, or the B gene. In hemp, hemp has an active CBDA synthase, and right next to it on the chromosome, an inactive THCA synthase. So we call that the BD locus, and those genes will predominantly result in the production of CBD and relatively little THC. In contrast, in drug-type cannabis, those genes are in an arrangement with an active THCA synthase and an inactive CBDA synthase. We call that the BT locus, and that results in primarily production of THC and very little CBD. Basic genetics, we all have 2 copies of every chromosome, one from our mother, one from our father. We have 3 possible combinations, or 3 possible genotypes then. True hemp would be a BD-BD homozygote and would have 2 active copies of CBDA synthase and 2 inactive copies of THCA synthase. That results in a ratio of about 23:1 total CBD to total THC, what we call chemotype 3. In contrast, on the other side, drug-type cannabis would have 2 copies of the BT gene. So it has 2 active THCA synthases and no active CBDA synthase. And we get about a 40:1 ratio, although I don't work on chemotype 1 plants, a very high ratio of THC to CBD. So that's chemotype 1, true drug type high THC. Now, if we were to hybridize a hemp plant with a drug type plant, we would get 1 copy of each, 1 active CBDA synthase and 1 active THCA synthase. And that results in these intermediate ratios, 1:1 up to 2:1, or maybe even 3:1 CBD to THC. We call that chemotype 2. Now, one of my graduate students, Jacob Toth, was able to look at these gene sequences and develop molecular markers to distinguish these 3 different genotypes from a very simple DNA extraction. And I'll show you those results in a minute, but what we have learned now, again, from sequencing drug-type cannabis, hemp-type plants, and CBD-type plants, is that those CBD plants are really a genetic hybrid between drug type and industrial hemp type. Because you can see in each of the 10 chromosomes here, we have a chimera, a blending of the hemp type genes in blue, in blocks of blue, and the drug type genes in blocks of red. And you can see like chromosome 6 here in this particular CBD line is entirely drug type, whereas most of chromosome 3 here is hemp type. Here's the critical part for CBD and THC production. A little block of hemp type, chromosome 9, interspersed in the middle of a drug type region of the chromosome. And also a gene here on chromosome 1, which gives us that very high overall total cannabinoid production. So these are data from C.J. Schwartz at Sunrise Genetics. So really what we want is then molecular markers that can distinguish this drug type THC gene from the hemp type CBD gene. And we were able to develop that and went back to samples from our 2018 CBD trial, which had 15 different varieties. And we extracted DNA from all 215 plants and tested the genetic arrangement in those plants. And you'll see that out of 215, 149 of them were true hemp type. They had the BD arrangement. But 64 were heterozygous, BD-BT, and 2 were true drug type, BT-BT. We also characterized the cannabinoid content in those individual plants and saw a very strong correlation between high CBD, 23:1, what we call chemotype 3 for the BD genotype, about a 2:1 ratio for the BD to BT, and again, that high THC, low CBD for this arrangement. When you look at the 15 seed lots that we had in 2018, you see that all but one of them had at least one heterozygote, and some of them had close to 50% heterozygotes, and those are surely going to go hot in your field. So here's a plot of the ratio of total potential THC on the vertical axis to total potential CBD on the horizontal axis. And again, we portrayed those chemotype 1 plants up here in red. The chemotype 2 plants are with green dots, with quite a range of both CBD and THC, but in about a 2:1 ratio here. And then the true hemp type plants with a range of CBD from 1% up to 13%. The critical thing to note here is this increasing slope of THC with increasing CBD, so that when you get to about 7% total potential CBD, that's when you cross the 0.3% legal threshold for THC. And you see pretty much everything that had a higher than 8% CBD production was above the 0.3% total THC limit. All of these data are in this paper here, which is open access, so you can go and read all of the original data if you want. The other thing we've been doing then is to characterize the genetic diversity of the hemp that we've included in our trials now. We're in our 4th season of doing field trials at Cornell. So we've analyzed the genomes of 192 different varieties, and they group out pretty well into particular groups. Everything on the left here are grain and fiber types. But you see there's quite a distinct genetic signature, for example, from Chinese fiber types compared to the European grain types or Canadian grain types. Everything on the right-hand side here are CBD hemp types, and they really do break out into about half a dozen founder lines or ancestral lines, including the Autoflower based on autopilot. And as you see, the Bayox types, Auto, T1 types, the hybrids from Oregon CBD, and a big group of cherry. So that's one conclusion. There are pretty much about half a dozen founder lines. There are variations within those lines. The other thing we learned is that as we collected samples from growers or from companies, the names don't often match the genetics. So here we have a cherry wine and super cherry, and here's a cherry by Bayox. And there may be some hybrids in here. Here's a Vermont cherry. So you can find cherry just about everywhere on this. Here's a cherry wine way over here. So if it's called cherry, I would be very skeptical that it really is genetically a true cherry type. And that leads us into our 2019 hemp trials. We had 30 entries in those trials planted in Geneva and Ithaca, replicated plots at each site. And this included some clones, some feminized seed type, and some dioecious types, which we sorted out the males using a molecular marker that we also developed for the Y chromosome here. So we planted in conventional horticultural spacing with raised beds and trickle irrigation. Again, one site in Geneva, one site in Ithaca, planted at about this week last year, and we're hoping to plant early next week. So we're pretty much on target. And we saw quite a bit of variation among those 30 types that we characterized. Again, 2 of them were day neutral or autoflowering from a company called Qiagene. Those types are now being marketed as Maverick and Pipeline, I believe. We just had a numbered ID for that. But these are very tricky to handle, I would say, in starting them in the greenhouse and then transplanting them. Our field wasn't quite ready when they told us we should transplant, which was at 14 or 15 days. We had to leave them a few days longer, and they actually flowered before we could get them transplanted into the field. So any little stress seems to be enough to trigger them to flower. They don't have a strict daylight requirement. We also had some types from a company called Industrial Seed Innovations, which I would say were early flowering. Unfortunately, even within the small plots that we looked at, we saw different flowering times for different plants within the plot. And I'll show you that in a couple of slides. We measured the height growth through the season, and my graduate student, George Stack, has broken down the growth curves here into different groupings. And you see this red grouping here, group 2, were a couple of varieties that grew very tall, and it's really impressive. They reached a maximum growth rate of over 3.5 centimeters per day and grew to over 2 meters tall. Some of them grew very early in the season, this group 3, and then their growth rate leveled off quite early in the season. But most of them reached their maximum growth rate in early July, maybe 2 to 3 centimeters per day in mid-July. As their growth is slowing down, then they're going to transition to flowering. That flowering is day length dependent for most of these lines. And again, what we saw was that some lines actually had early flowering type plants mixed in with later flowering type plants, as much difference as 3 to 4 weeks. So the dark green blocks represent the date when we first saw terminal flowering for each variety, and quite a range from flowering at the time of transplant to flowering as late as 16 weeks after transplant. The little white asterisk here represents the sample that actually went hot for total THC. So you see some of these varieties are exceeding the 0.3 limit even 2 weeks after the onset of flowering. Let me show that in a little bit different format here. So what we've got here is the planting date, and then the bars represent the average time of the start of flowering. The triangles represent the point in the season of maximum growth rate, and then the X'd out circles represent the time when that sample exceeded 0.3% total THC. So for all of these varieties, we started sampling shoot tips at the onset of flowering and then sampled weekly for 6 weeks after that point. What you'll see is there are some types that never went hot, that never exceeded the 0.3% threshold. There's some that, again, the production of cannabinoids was so rapid after the onset of flowering that they went hot even just 2 weeks after the start of flowering. So what George has done is to graph the rates of production of total potential CBD and align all of these graphs according to the first week of terminal flowering. And we do see a few different patterns in here. We see some that just rapidly increase and never seem to plateau in their production of CBD, achieving over 20% CBD. There's some that have a moderate rate and then they level off, and maybe we even see a decline in cannabinoid production as we perhaps let those flowers stay in the field a little too long. So you see a little bit of arching down here. And then some that start early and produce cannabinoids at a very modest rate and never exceed the 0.3% threshold. So here's the same type of curve for production of total potential THC. And if I can flip back and forth between these, you'll see that they align very nicely, right? So the curves for the production of CBD match those for the production of THC very closely. The dotted line again represents the 0.3% total potential THC limit. And most of these lines are hitting that after 2 to 3 weeks. There are some differences in the timing of when different varieties cross that threshold. So that's something we'll want to characterize better over time because some of them are a little bit slower in their production of THC. So that gives you a little bit more time in terms of getting regulatory sampling done. Of course, the other key aspect of production is the biomass yield. And so for each plot in each of the trials, we cut and dried and stripped an entire plant per plot. So a total of 4 plants for each site. And well, first of all, you can see the Geneva site, which is our McCarthy farm, out-yielded the Bluegrass Lane farm in Ithaca pretty consistently for all of these varieties. Some of them had more than double the biomass in Geneva than in Ithaca. The soils in that farm in Ithaca were actually quite acidic, quite low pH, which I think is probably a major contributor. And we also had soil moisture sensors in the field in Geneva that I think allowed us to control our irrigation more precisely than on the Ithaca farm. Despite these low yielding day neutral types, those are included in the averages. We averaged 2 pounds per plant in Geneva and about 1 pound per plant in Ithaca. But you see the maximums here were over 1.7 kilograms. So between 3 and 4 pounds per plant in the high yielding varieties. So then you can multiply the biomass yield times the CBD percentage, and get your total yield of CBD per plant. And that's really what we're looking for here, is a combination of biomass yield and overall whole plant CBD production. So that's the next question, and something we're going to focus on this year is when we looked at the concentration of CBD in that final shoot tip sample and compared it to the whole plant strip sample, we saw quite a range in the ratio of CBD in the shoot tip to whole biomass. So we really want plants up here where the whole plant biomass has an equal concentration of CBD to sampling that optimal shoot tip. But we do see some lines where the concentration in the shoot tip is double what it is in the whole plant strip biomass, or to put it the other way, the whole plant biomass has half the concentration of CBD as that optimal shoot tip. So this year we're going to do a much more careful evaluation of canopy development, of branching, and specific sampling of shoots in different parts of the plant, and try to learn a little bit more about the variation in cannabinoid production according to canopy form and shoot location on the plant. We also saw quite a bit of variation in powdery mildew in that trial. So quite a bit of powdery mildew in our McCarthy trial, but we did see one variety here, First Light 58 from Sunrise Genetics, that had essentially no powdery mildew incidence either in Geneva or in Ithaca, compared to some which were really quite covered with powdery mildew. My wife, Chris Smart, is now working on controlled inoculation. She has a number of isolates of powdery mildew that she maintains, and a very good system of inoculating them in a mist chamber, and then evaluating the resistance or susceptibility of different lines after that controlled inoculation. Right now, we have made some crosses with First Light 58, which we think holds a resistance gene with some other susceptible lines. And we're now surveying those F1s to make sure that we can identify a plant that has inherited that resistance gene, so we can go on to characterize that gene and develop some markers to introgress that resistance gene into other commercial lines. I'm going to run through this really quickly. That it does lead us to the question then, if there is any impact of disease, for example, or other stresses on CBD production, or on the ratio of CBD to THC. So last year we set up a trial specifically to test this with 3 different varieties. There were 3 plants per plot. So in each treatment block here, a total of 9 plants. And we applied Ethifon, which releases ethylene, which is a stress hormone. It also promotes female flowering. I wounded the plants with a weed whacker, flooded them by putting a hose into those individual plots on a very regular basis, and then sprayed them with 0.5% Roundup. And Chris also inoculated some plants with powdery mildew. So the bottom line, again, we sampled at the time of the treatment of these different stresses, and then 3 weeks after that, every week for 3 weeks after that. The 3 lines that we used included one of our breeding lines here, 191039, a T2 line from Boring Hemp, and a clonal line TJs from Stem Holdings in Oregon. What you see is that the only significant difference by treatment was a lower level of CBD in the herbicide treatment. And really that 0.5% Roundup was a little too high, pretty much killed the plants. And by the end of that 3-week sampling period, it was hard to find any green tissue left. No significant difference in the amount of CBD among any of those other stresses. And this is the time series. So again, you see a progressive increase from that time 0 when I applied the stress to 3 weeks after. This is the average of all 3 varieties here. And you see they were getting up to 10 to 12% CBD at the end of that 3-week period. And clearly a significant decline in CBD production due to the herbicide treatment, but no other significant difference based on stress from the control here, from the black bar. And then the final question is, would stress cause a difference in the ratio of CBD to THC? And again, this is by variety. We don't see any significant difference in the ratio of CBD to THC. They're all pretty close to 25 to 1, and no difference based on the stress treatment. So if you hear that stresses, and at least these were the stresses that I was able to apply, flooding or stress hormone or powdery mildew or wounding don't seem to have any effect on the ratio of CBD to THC. The only effect they might have is actually to kill the plant, which would reduce your cannabinoid yield. One final point, again, something we're going to work on this year is I think we're going to end up with about 10 CBG lines, and we will have a specific trial to compare CBG, including white CBG from Oregon CBD. That is what we call chemotype 4, and the high CBG production results from a total lack of any of these genes. So basically either a knockout of the THCA synthase or in a drug type plant, or the knockout of a CBDA synthase in a hemp type plant, or a knockout of that entire portion of the chromosome. So we're gonna evaluate some of those CBG lines. Just a caveat here, already in our hands, the line that we've gotten is quite unstable. So just be careful. You might want to keep these CBG lines separate from your other CBD lines in case they do begin to produce male flowers or perish the thought they might go hot. So I will end there. Look forward to questions from the group. But I need to thank our funding sources, New York State Ag and Markets and Empire State Development. But much of this work was funded by a sponsored research agreement we have with Pyxus International, which is a large crop management company in North Carolina. We have a lot of material on our website, and although we haven't gotten due to our research pause, we haven't gotten our 2019 reports up yet. We will have them up very shortly. And don't hesitate to follow me on Twitter @CornellWillow. So I will stop sharing and be happy to take any questions.
Okie dokie. Well, thank you very much for that presentation, Larry. It's quite interesting to really dive into all the genetics of it and get a better understanding so that we can potentially start with genetics and then figure out, predict what's going to happen later on so we don't find out after we're going to harvest the crop. So we have one question so far. So now they're starting to come in. Let's see. Elram AR asks, in your video, will you test heat/drought as stresses that affect cannabinoids?
Yep. So that is a great question. Actually, one of my treatments started out being drought. And drought experiments in New York mean that you have to have a rainout shelter. So I tried to build rainout shelters with plastic tarp and pipe. And the first weekend we had a storm and they all blew down. So it is very challenging to do drought experiments in upstate New York. You have to be able to control the rain. If I were in the Central Valley of California, that's pretty easy. Heat is another one that's hard to control, except in a greenhouse or a growth chamber. Actually, I would point you to one of my colleagues at Utah State, Bruce Bugbee, who's done some great work on that. So I think we're going to learn more about the impact of heat and drought.
Cool. Thank you very much for answering that. So Joseph Calderon, I apologize if I'm mispronouncing these names. Any correlation between stressors like wounding and terpene expression?
Oh, Joe, that is a great question. So you notice that we have not had the bandwidth to do terpene analysis, but I have seen a lot of work from Sunrise Genetics in Colorado. They've done full terpene analysis in their field trials. And unlike the cannabinoids, which seem to be quite stable in different environments and in response to different stresses, the terpenes can vary much more. So I think there will be much more effect of stress and environment on different terpene levels than there will be on cannabinoid levels. Great question.
Cool. Yeah, absolutely. Let's see here. Next question. Edmond Chabot says, hello, and thank you for the presentation, Larry. Will there be any in-depth research on grain and fiber varieties this year? I am a grower in Madison County. Thanks.
Yep. So again, this is our 4th year of doing grain and fiber trials. We planted our grain and fiber trial in Geneva today. So I'm in here giving this presentation instead of out there helping with the planting. Unfortunately, because of our research restrictions, we will only have trials in Ithaca and Geneva. Last year we had trials up at our Willsboro farm, our Aurora location, and our Freeville location. So those trials are scaled back to just Geneva and Ithaca, but we will continue characterizing those grain and fiber types.
Very cool. So Michael Casper asks, hi Larry, and any comment on the new CBG cultivars we're just out in 2000 white CBG from the folks at Oregon CBD.
Yep. All I can say is that we have the seed planted, and we're going to put it in our trial, not next week, but the week after. And like I said, we've gotten, I think, 8 or 9 other quote unquote high CBG lines from different sources. And as I mentioned, I think there are 3 sources of genetics to produce that high CBG: knocking out THCA in a drug type plant, knocking out CBDA in a hemp type plant, or knocking out the entire gene. We have a line called Xantheca that our molecular markers don't work on that. So we think that's an example of the total knockout of those genes that produces the CBG. So we are trying to breed our own high CBG and CBC lines and learn about the functions of those enzymes along the way.
Ulrich Sitter says, what is the latest on oilseed / food grade hemp trials? Any organic seeds available? It is hard to find organic, local food grade hemp.
Yep, Uli, I mean, you and I have communicated quite a bit about this. I actually have, I'll just say right now, I have quite a bit of seed that didn't get planted last year. I have Biala Brzeski, I have Anka, that were supposed to be used for certified seed production last year, and that didn't work out. So if there are organic growers out there, please come and take the seed out of my freezer. You know, you just need to assure me that you're not going to propagate that seed next year, that you would just use it, sell it to Uli to make her delicious organic hemp seed oil.
Since you are so entwined in genetics understanding on this, roughly on average, how long does it take to take an unstable genetic and stabilize it?
Yep, that is a great question. And there are different strategies for doing that as well. All of the grain and fiber types so far are populations. So every year as you produce seed, you're allowing dozens or hundreds or thousands of individual plants to cross with each other in a population. Many of these CBD lines are inbred lines. And so there's a good question of how many cycles you can inbreed before you see inbreeding depression. But I think what is the growing consensus is that the most productive and most stable lines are true F1 hybrids. So they're the result of the cross of two inbred lines. And I believe that's the basis for most of the lines from Oregon CBD. And that's how they get that good uniformity while also getting high production and all of the traits that they want. So we're looking at probably 3 to 4 cycles of inbreeding, and then some discovery of how you can generate the best F1 crosses. So really it's going to be a process of probably 4 to 6 years
before we really know.
Before we really have good, sufficient field testing data to validate the traits that you're promoting as being unique.
Yeah.
Right. So there are 3 characteristics of a new cultivar for any plant type. It's distinct, unique, and stable. So you need to show whether you're applying for plant variety protection or for certification through AOSCA, that your new variety is distinct from everything else, that it's stable, and uniform. Distinct, gotcha. Uniform, and stable.
Yeah, gotcha, interesting. And in terms of the F1 genetics, is that the one of those 12 original that you were talking about?
So those are really the founder sort of inbred lines.
Gotcha.
So I think what we're seeing is, for example, an F1 cross between a cherry type and an auto type. And that might give you some hybrid vigor, but that F1 is also going to be very uniform.
Gotcha. Okay. And then Mark Gherkin asks, what emitter spacing and GPM are you using for the drip lines? Thank you.
Yep. We're just setting up our trickle lines now. So we're using 12-inch spacing. I think it's 0.45 gallon per minute per 100 feet. Pretty standard. It's pretty much the same thing that my vegetable colleagues are using. And then we're injecting soluble fertilizer, conventional fertilizer, through a dosatron unit based on leaf color measurements that we take through the middle of the season.
Gotcha.
And so you can measure the green color of the leaves with a little clamp-on sensor. And if they start to go a little bit yellow, it's a good time to give them some more nitrogen, and they will suck that right up and turn dark green within a couple of days.
Cool. Awesome. Yeah, nitrogen seems to be the key.
Yep.
So CJ from Growing Renaissance LLC asks, for greenhouse planting, once plants are flowering, does light modulation control CBD and THC percentage?
Yeah, that is another great question, and it's a little bit out of my area of expertise. But fortunately at Cornell in Horticulture Ithaca, we have an expert in greenhouse lighting, Neil Matson. So Neil has an experiment going on right now with LED lighting with different ratios of red and blue light. And then he will measure the cannabinoid production at the end of the season. He's also going to be looking at some day neutral types, autoflowering types for greenhouse production in the summer in New York, so that you could get production in down times without relying on natural light, right?
Gotcha. Cool. Thank you. Elram AR asks another question. Will Cornell be posting information on pesticides available for New York, or for hemp in New York?
Yes. So actually there's quite a comprehensive list of what has been approved by New York State DEC. And we have a link to that and sort of it's a little bit tricky how to do the search in the database. So if you go to our website at hemp.cals.cornell.edu, we have a link to how you can get to the New York State DEC website. You search for cannabis, and I think the last time I searched, there were more than 75 products that were approved for use on hemp in New York State.
Wow, cool.
Most of those are biologics.
So Melissa Gibson asks, what was the most unexpected and surprising finding in your years of CBD research?
Yeah, so I'll say the surprising thing was the high percentage of non-compliant plants that were being sent to us as hemp. Our legal counsel doesn't really appreciate that. But really, scientifically, the thing that is most interesting to us right now is the control of flowering time and sex. So hemp is quite fickle, I would say. It's quite easy to turn a female into a male, and that's the way we produce feminized seed. So it makes you wonder what the role of the Y chromosome is there. If you can get pollen and male flowers without any Y chromosome present. And likewise, in the monoecious grain types, those also have an interesting mutation and no Y chromosome. So we're really interested in trying to learn more about the mechanism of sex determination in hemp, and also get some good tools for managing flowering time.
Gotcha. Very cool. LRAM AR asked another question. Great question so far. Have you studied the effects of different mulch materials on hemp growth?
No, we have not. Again, I only have so much bandwidth, so we've tried to stick with pretty conventional cultivation methods. This year, we will be doing direct seeding of CBD types, though. That's a little bit challenging for us because the way we would normally do that, the way my vegetable colleagues do that, is to load up a monosem planter, but to convince companies to give us enough seed to get that monosem planter going, we haven't been able to do that. So this year we bought a little walk-behind seeder, and hopefully that will be a good simulation for being able to get plants direct seeded. We'll use overhead sprinkler irrigation instead of trickle. And yeah, I was hoping to do a comparison of plastic mulch to no plastic mulch, because we also just obtained a nice Chichi-Magli transplanter. But that's one of the things that I had to cut back because of our COVID-19 safety restrictions this year. So
in terms of the production of CBD particularly, or other cannabinoids, and it seemed to kind of correlate, is higher THC production kind of necessary for higher CBD production, or does it seem like you can kind of split it off?
Yeah, so we're pretty confident based on reading of the literature and the results we have from close to 4,000 samples that we've run so far, that CBDA synthase makes CBD and THC. It makes CBDA and THCA, and pretty much in a fixed ratio of 23:1. Wow. So I would be very skeptical when you see a company promoting, for example, a 30:1 or even higher ratio of CBD to THC. Biochemically, it's not what we've been seeing. So that enzyme, CBDA synthase, is a little bit leaky. It makes mostly CBDA, but it makes a little bit of THCA at the same time. So the more CBDA you make, the more THCA you're going to make.
Gotcha. Interesting. So CJ, he keeps on adding to his name again, asks, or CJ Seagull Isaacson, I'm sure he just wants me to get his name out there, from Growing Renaissance again, asks, what do you think is the amount of hemp being grown in New York State given a difficult harvest for New York State farmers in 2019?
Yep. I don't know exactly, and we don't have all the planting reports submitted to Ag and Markets yet, so I don't think they know exactly either. Just from what I've heard, for example, from the seed companies and from producers of transplants, sales seem to be down. Unfortunately, I've been cooped up here at home, so I haven't been able to get out and talk to too many growers. But it does seem like there is significantly reduced acreage compared to last year.
Yeah, definitely seems to be very much the case. It's people got scared, which is understandable.
But I will say that Ag and Markets continues to get new grower applications. So I think we're up over 700 licensed growers in New York, and they continue to process new applications every week.
Yeah, so new people are wanting to dip their toes in the water and kind of see where they can take it. So another question from here. So kind of on that note of THC production being very much correlated with CBD production, I mean, I know it would take mass amount of change for the legislation to change, but is the 0.3% limit kind of, is it a realistic expectation if we really want to seek the best genetics?
No. And we have a quote from Professor Ernie Small at University of Toronto. He sent me an email. We've asked him. We all know that he was a key player in helping the Canadian government establish that 0.3% limit.
Interesting.
And you can read his explanation on our website on the FAQ page. Really, that limit is to distinguish hemp type plants from drug type plants, right?
Gotcha.
And I've showed you pretty clear genetic distinctions between hemp type and drug type. The true hemp type plants rarely exceed 1% total THC. Even the highest producers of CBD, you'd have to be up to 23% CBD to get to 1% THC. And that's pretty high.
Yeah, that's pretty high.
Whereas the drug type plants nearly always are above 1%.
Gotcha.
So just based on our data, I would say that good round number of 1% is a pretty clear threshold between true hemp type and drug type.
Yeah, absolutely. So kind of to jump along that note, is there a limit of total cannabinoid content within a plant? Like I'm sure at some point in time, the plant would just become pure resin and it would just fall apart. So
yeah. So that's a great question because it depends on how you're going to deal with that. So if you were growing individual plants that you were just going to clip by hand and use in a very, carefully manipulated way, a high percentage of 23% would probably be okay. But try to harvest that with a combine. You'll probably get about 100 yards and your combine will be totally coated with sticky goo and it'll get totally jammed up and nothing will go through it. So there's also a practical consideration of how much resin can your mechanical harvesting and processing systems actually handle.
Absolutely. Cool. So yeah, so it's not just the maximization of the plant itself, but it's also the understanding of the whole supply chain and how that's going to affect the whole thing. Cool.
And being able to do it efficiently. Yep. So it really depends on how much mechanization you want and what your end product really is.
Gotcha.
Yep.
Very cool. So I just have one more question. So you would on that we have about 5 more minutes, and then we'll wrap up with this. On that note where you're showing the wounding, pesticide use, and all that kind of stuff in terms of CBD production, it seems that pesticide or specifically Roundup was the only thing that really knocked everything off cue. So in that case, is there an alternative that you could use for pest management, or is it just a matter of we just don't use it at all in terms of like getting it up in numbers?
Maybe you could restate your
Yeah,
question.
so
I pretty much killed those plants with Roundup.
So I mean, I'd imagine it, part of it just because they're not Roundup ready plants, so that would definitely make sense in terms of that aspect. But is it the matter that like are there alternatives for pest management that we could use in terms of herbicides, or is it just an option that we just choose not to use any herbicides or pesticides at all?
Yeah. Okay. So that's really, I think it's a two-part question. So in our grain and fiber trials, which we just finished planting, we will be doing a research application of herbicide. We're going to try Prowl H2O for weed management in those grain and fiber trials. And that's based on a few years of research at North Carolina State, University of Kentucky, Western Kentucky University, where they have been doing herbicide trials, and also the advice of our weed scientist, Lynn Sosnowski at Cornell. Now, whether that's a good idea for producing CBD extract is another question, because those CBD extracts will be tested for pesticides, and some of those pesticides may end up in the CBD concentrate, whether it's just through contact or whether the plant is actually taking it up and accumulating it. I'm not sure about that yet, but even spray drift of pesticide from an adjacent field may end up getting concentrated in your CBD extract, and then you fail the final pesticide test in the extract. So in that point of view, it may not be worth the risk to even try using herbicides if they're potentially going to end up in your CBD extract.
Gotcha. Do you think you have time for one more question? Just one popped up here, and then we can end it there. No more questions after this. So Elmar AR asks, has Cornell done any work with post-harvest chemical changes in cannabinoids?
Yep, that's another great question. I think there's a lot of opportunity to look at different storage conditions. I know that last year people tried to do some baling. I'm not sure how well that worked out. I know that some growers had access to controlled atmosphere storage conditions that we would normally use for apple fruit. So yeah, I think that's a great opportunity for additional research to try to optimize the long-term storage of flower material, cannabinoid material. I can tell you that, again, last year we analyzed about a little more than 2,500 samples. So we had to dry all of those samples. And sometimes our drying rooms overheated. And when that happens, you see a clear shift from CBDA to CBD, and unfortunately from THCA to THC. And because New York State is only looking at the Delta-9 THC, that causes your samples to go non-compliant.
Gosh, yeah, all the challenges that we have to deal with.
But it's also an interesting question. I don't know what the conditions are for the conversion of Delta-9 to CBN. CBN is a breakdown product of Delta-9 THC. Yeah. So if you had a post-harvest issue with high concentrations of Delta-9 THC, are there some conditions you could apply to convert that to CBN? That's a great research question.
Absolutely.
We'll
have to look into.
Hopefully we can work on. Yep.
Yeah, cool. Well, thank you very much for joining us, Larry. It's been a pleasure to hear about your research, and you actually kept right within that half-hour mark. It was perfectly spot on. And thank you very much for helping us represent Hemp History Week. And we're going to continue on for many years that the hemp industries are going to help support us here in the United States, in New York specifically.
Yep. let me just put
Yep.
out sort of a save the date. I don't think we're going to be able to have an in-person field day this year because visitor access to Cornell is restricted right now. So we are going to try to hold a virtual field day, and that will be on August 20th, Thursday, August 20th. So mark that day on your calendar, and we're going to be collecting video through the entire season and try to package that into some presentations in combination with some live presentations and Q&A sessions. So
awesome. Cool. Yeah, all the adaptation that needs to happen.
yeah.
So thank you everybody for joining us, and have yourselves a wonderful evening, and we'll have to talk again soon. Thank you for joining us on the NYHIA network. If you enjoyed the video or you found it helpful, please like and subscribe below. If you're interested in growing the hemp industries here in the United States, please join us at the HIA at joinhemp.org, and make sure you check off your state chapter during sign up. If you want to find any of the links that we have below, they're in the description, and please comment to let us know what you want to learn next. Thank you very much for joining us, and have yourself a wonderful day.